There we are. Hello. Uh, Hopefully my internet seems a little bit weak at home today, everybody. But this is the review recording. And again for the new students, I just do this. I basically am narrating. I'm looking at the exam, and I'm just trying to sort of give you a verbal study guide in addition to the one that was that's on Blackboard or I sent to you via Blackboard. Nevertheless, it starts here. I mean, and again, these are I think it's 70 questions, multiple choice, true, false, matching. Typically, the matchings, it's used once, twice, or not at all, and this is typical of them. It starts, and again, it's something we're going over tomorrow. These are the neurotransmitters, and basically, the key involves different. It's by category, whether they're whether they're amino acids, whether they're biological means, whether they're amino acids, whether you're going to see there's gaseous agents, whether there's enkephalins is another one that's in there, and you'll see all of these. So we do it by category. And then what I basically do is give you a nugget for each category. And since there's a lot of number, like for instance, catecholamines, and I just started to touch on it, those biogenic amines all come from tyrosine. And that was dopamine, norepinephrine, and epinephrine. Those are all neurotransmitters. The epinephrine is the oddball because it's produced in the adrenal medulla, which is really sort of an out of position part of the nervous system, even we consider it an endocrine gland. It's kind of both. And I find that to be pretty interesting as well. So that's one aspect that we could, that you should be aware of. And we talked about indolamines just a little bit. I'll review it. Like, which had histamine, made histidine, and tryptophan, which made serotonin. Those are significant. So I mean, those are the little nuggets that you would have to match up in one position or the other. We haven't gotten to gaseous agents like nitric oxide. We haven't gone into enkephalins, which are morphine-like substances and agents that are similar, which are small polypeptide chains that deal with pain endogenously, which are produced inside of you for us to deal with pain. Just some examples of some of the things that you're going to be running into. You got to be familiar with the standard G protein passageways versus the ones that where the steroidal agents make their way through the membrane, the G protein. Understand that's the primary messenger is whatever the hormone is, whether it's growth hormone, whether it's insulin, whatever it might be that would trigger that. We'll go over so many of them. And then that's the primary messenger. So cyclic AMP and calcium are the secondary messengers. So I think you have to be familiar with all of those. I mean, they're so this is a typical pathway to know the distinction between G protein receptor use and then how the steroids work, which pass through the membranes of the steroidal based triggering agents, hormones, chemicals, whatever you wish to call them. They reach receptors in the cytoplasm, which are then make their way by way of that receptor uh, ligand uh, unit that forms. I guess that I'm trying to think of the best word. I don't know if there is such a good best word for that. And then subsequently, what it does is it passes through the nuclear membrane and basically it acts itself through transcription and translations, where the G protein typically work their way through those second messengers, which trigger cascading reactions, a la tyrosine kinase, which you're probably pretty familiar with from the usage of insulin. So those are things that are important as well. Cell cycle is a big topic, and this is from the initial chapter. The first one was a little bit out of sequence, but I just stuffed it in there. Uh, know what goes on in G1, G2, and S. G1, really doing most of the work, and really maybe the most important and the most active phase from a standpoint of getting all the materials and reproducing all the building blocks to make everything ready. All of those, basically putting everything into play that's going to be needed, with the exception of generating, of rather duplicating the DNA, which is all about S phase. That's where mutations occur. And then, of course, G2 is final preparations. These making sure that now that everything effectively is complete, is it effectively ready? Do we quote unquote kind of have this chemical go ahead to proceed with mitosis and then finally cytokinesis? So the G1 phase is all about making sure that, let's say, think everything is the building blocks and all the necessary materials are there. S begins the assembly as well as G1 and SB is the assembly rather of DNA. G2 is that final inspection, if you want to think of it in a simplistic ways, and it moves on. And remember, they're based on the cyclin, and cyclins to me are like a waste product. There's something that they're a chemical that is produced each time it undergoes mitosis because it's destroyed at the end, and it bonds to these cyclin-dependent kinases, forming a complex, and that's what clears those restriction points, those basically blockers for the cell cycle proceeding. And again, to me, it's just the idea of governance. In living systems, we govern, we hold it back from going too fast, effectively. So it's like a brake governor that a big truck would have going downhill. Uh, the type to keeping your engine from racing all the time. However, whatever way you want to view it, and biological systems are like that. Heart rate is a perfect example, because you're going to see down the road that acetylcholine generated by the vagus nerve keeps our heart rate in check, which is desirable because it would go too fast otherwise. So a lot of that occurs. So the cell cycle basically is about a lot of problems. And so as we look at these things, and my spelling is atrocious, I think sometimes, but we're okay. So you look at all of these things, and the answer to most of these, rather when there's errors that are made, is effectively one of several things. Theoretically, a cell might reach a point where it goes to G sub zero and it exits the cell cycle, but most of the time, those errors result in some degree of apoptosis. There's some autophagy. The cell destroys itself, gobbles itself up. I'm not getting into why one occurs and not the other occurs. I think the mechanisms are may not be different, but the end result is certainly similar. So I think all of those things are part of it. So I think that's all stuff that you need to remember. And then like it's so easy. I mean, you have two parts of the cycle where effectively nothing happens that other than one event in prophase, metaphase, anaphase, and telophase. Metaphase, they line up in the middle, and anaphase, the shortest phase, they start to separate. Prophase, four events, major events, not worried about early, late, or in the middle, or wherever it occurs, or which one of these occurs first. The chromosomes appear because they begin to supercoil as they move away from those histone proteins. The nuclear membrane appears to disappear. It's still there, it's just disassembled. The nucleolus disappears, again, it's disassembled, and we begin to start the spindle. And those four are reversed in telophase. Somewhere in the midst, somewhere at the very end of anaphase or during telophase, we begin to see cytokinesis. And that's highlighted by, and again, we're talking about our cells. We're highlighted by that cleavage furrow. Those underpinnings begin to, they're really actin filaments that are beginning to exert a force and starting to increase the degree, almost this crease. And that's really what the cleavage furrow is in between the two cells, which eventually will, because of the inherent, not flexibility is maybe the wrong term, that sort of interesting feature about membrane that can assemble or disassemble since it's not bonded together. It's just they adhere to one another and different signals can cause it to separate. So remember all of those. Okay. That's what I mean. And again, it's a trip up type of a question. If you see it, I don't even know if there's one on here. Mitosis is nuclear division cytokine. I mean, cyto, mitosis is nuclear division. Cytokinesis is cellular division. So a lot of times that's a trip up question, particularly going forward on other kinds of exams. Look for different because it's mitosis. The nucleus divides, the cell divides. There's those subtle distinctions are really little interesting separators. I know that you would classify them as trick questions. The nature of life is trick questions. There's a lot of tricky situations. I don't mean to get overly philosophical about it, but that's exactly what's happening. Okay, you have little things where a tiny subtle decision can really impact what's going on with you. So I ask you to be wary, and I warn you to be wary, and not just from my perspective. You can yell at me. So moving on. You know about exocytosis and endocytosis, that they're forms of active transport. You're familiar with that. The neat one to me is the receptor mediated endocytosis. It's one I talked about when people have these hereditary lipidemias, because these typically low density lipoproteins are unable to enter the cell, so they stay in the bloodstream where they are much more likely to cause problems. Okay, much more likely. And what occurs is if they are out in the bloodstream, then you have the potential for adhering to areas of inflammation and damage in the intravascular environment, and eventually leading to fatty plaques and then what we call hardening of the arteries. We use technically it's athero because an atheroma is a fatty compound, but they're effectively arteriosclerosis, sclerosis for hardening arteries, etc. It affects more than just arterial blood vessels, by the way. That's why the athero is a better name. We're not going to really deal with that. I'm just sort of giving you again additional context for this area. But in the absence of those receptors in those clathrin coated pits, what happens is they can't be brought into the cell where they can be utilized. Then we can utilize these low density lipids, the cholesterol molecules, efficiently and appropriately because they play a variety of roles in the cell. If they're stuck in the bloodstream in excess, that's when there's problems. And that's really, there's a lot of science behind it. Again, we talk about it in the heart. That's the whole basis of what's called the Framingham study from 1980 when it was published, and stuff that is used all the time today. When you get older, you reach a certain age, and hereditary family history rather plays a role, you start doing lipid profiles. You have to fast overnight, da, 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 da. You know family members this has happened to, I'm sure. Okay. Remember all the cell adhesion molecules, the different roles of desmosomes, tight junctions, and gap junctions. Enough said. I'm sure you've gone over it 1,000 times, but be familiar with it. Okay? And as I made a special note. Concentration may drive the bus when it comes to diffusion. Okay. But particularly when we're going to be crossing membranes in simple diffusion, permeability plays a pivotal role. You'll see this really manifest itself when we start talking about respiratory physiology. So if the most important is concentration gradient, but understand getting through the membrane, not all things get through the membrane equally if they are soluble. It's the degree of solubility. The role of the sodium potassium pumps we're so familiar with. They're really an example of primary active transport when we couple other things to them and utilize that energy or that flow. Okay. So we take that pumping mechanism, we either move things in, co-transport or symport, or we move other things out. We antiport, and be familiar with that. Okay, let's see. Okay, and I think my spelling is just terrible. Ah, yay! I think I corrected it. I'm looking at it. It has those little grammatical corrections, and I was guilty of one. So as we move on, and hopefully the dog doesn't interrupt us in this particular narration, that we look at different parts of it again. When you're talking about endo and exocytosis, the whole idea is this remarkable ability for basically the membrane to open and close. And to a certain extent, it's almost like amoeboid motion. We're either creating those pseudopods or we're sort of utilizing them in reverse almost. And so both of those are interesting, and typically that is a responsibility of microfilaments more than anything else. That's because remember, those are the structures that are most frequently found and are really the byproduct of like the cleavage furrow and things along those are the roads where microtubules are much more specific to things like flagella and cilia. So it's a little bit different. And plus, they have a lot of internal roadways with microtubules. The actin filaments are more under the cellular membrane, so they're much more significant with regard to uh, endocytosis and exocytosis, which really okay. And remember what the snare proteins do. We talked about it. We talked about the relationship to Botox, which processes that you should know it off the top of your head. We look at it extensively in the neuromuscular junction. We look at it with exocytosis of neurotransmitters. We just did so recently. And resting membrane potential, such as the roles of sodium and potassium, are important. I think all of that's relevant to you. As we begin to move on to the basically looking at muscle physiology, again, another one of these matchings. These are, and I'm happy to share the answer key with you: tropomyosin, troponin, G-actin, calcium, and titin. And so imagine what the questions would be from that. I mean, you have to know the role of tropomyosin, what it covers, what type of, how it is arranged. Is it a plug? Is it a thread? What holds it in place? What releases it? Okay. What, what's the construction of actin and myosin? All of those things are important. What creates the A band and the I band? What kind of overlaps? Where we see them, what connects them to the Z disc? Are they directly connected or are they indirectly connected? Those are all the kinds of things. Okay. How, and then basically taking it from the impulse, I'm going to move this muscle. Go ahead and start there and think of what happens to that nerve. The impulse goes down. It triggers a voltage gated channel. What kind? Then it facilitates exocytosis, secretory vesicles, the neurotransmitters, specifically acetylcholine, the role of acetylcholinesterase, what role, the significance of that. What are the things does it do? What are those three things? Diffusion, breakdown or metabolism, reuptake or resynthesis. And again, because it's a really good introduction to the role of the nervous system. So that's all of the significant aspects that are there. What happens at the motor end plate once that acetylcholine goes? What does it contact? What kind of channels? What moves where? Where does sodium move? Where does potassium move? What does it create? How does it create that action potential specifically? I think those are all types of important questions that are due that you have to be familiar with. Remember the 2-to-1 relationship in skeletal muscle, and when we get to smooth muscle, the 12 or 13-1, depending on the author that you read, relationship. That's why I phrase it that way. What's contained within that sarcomere? We know we have mitochondria. How is it? What carbohydrates do we use for energy storage? What protein holds on and kills available oxygen? And don't forget all of those things that it can do. The fact that it starts anaerobically. Remember how much effectively inherent ATP you have available. It's very limited, but most of the activities we do, we replenish it right away. They don't take 4 to 6 seconds. They take a fraction of a second for me to move my lips, for me to speak, for me to gesture with my hands, which I do inevitably even when I'm at home. All of those kinds of things. Fractions of a second or a second or two. We replenish that ATP after that. But if we have to go longer, now you have the creatine phosphate pathway. And maybe you have another technique, and again, it's another high energy phosphate that we can sort of flip flop and magically recreate, add that phosphate to ADP and get ATP. Well, that's all good. But once that's out, what do we do then? Then we have to go into the anaerobic pathway. It's glycolysis. I don't have to reteach glycolysis. We know the problems are the development of lactic acid, which is going to go through the bloodstream, although it has uses. Eventually, it's going to have to be dealt with. We create the lactic acid because, in the absence of oxygen, the parking place for those two little electrons from that one NADP or H or whatever the hell it is, but you know what I'm talking about, got no parking place. Oxygen is the parking place. In the absence of oxygen, we only have the substrate left. So what happened? Pyruvic acid, rather than being able to enter the citric acid or Krebs cycle or tricarboxylic acid cycle, what does it do? Unfortunately, they park in there and it makes lactic acid. And eventually, when we replenish the oxygen, the liver is able to convert that to pyruvic acid so it becomes useful. And so all of that, all of those, the anaerobic glycolysis event, by this time, maybe where are we? 45 seconds to a minute of sustained muscular activity. At that point, if something is not done, the waste products alone will begin to create damage. They're mostly ionic products. The lactic acid is less of a culprit than it is the ionic products and the waste products that cannot be filtered out because the muscle is contracting so rapidly. The blood supply is cut off. So you can't bring more blood in and you can't get waste products out efficiently by the venous system. So, I mean, it's fairly, this is a well understood structure. It's a well understood mechanism rather than structure. Both are well understood, obviously. That's all part of it. So by that time, you better convert to aerobic. Aerobic just means basically you have to be utilizing large muscles. You have to basically adapt your breathing and heart rate. to what the usage pattern of those muscles are. And once you do that, you can go for significant periods of time. We all can. It's no magic. I mean that, and really that's where it becomes so impactful in cardiac fitness, which is where I should be going tonight, but I'm not. And then the whole thing about stimulating those muscles, we do it in the lab with the isolated twitch. So remember, if that we did 1 out of 10, that was an isolated twitch. If we do it more frequently than that, let's say 20 times a second, now you get this phenomena called staircasing because there's still more calcium. The calcium is being pushed out, not back out, additional calcium before it's pumped back in, and the muscle has a complete chance to, and the twitch has an opportunity to resolve and return to resting. If we do it faster still, we get that unfused tetany. And if we get up to like 50 a second, now we get that fused tetany. And most of the time, we're using these techniques in electrical stimulation and physical medicine and rehabilitation. As I said, and some of the athletes in the class can basically point out their experiences because certainly they've had them. This is used all the time, and it's been around for a very long time. It's been under a variety of different names over the course of time. So let's see what else. Now, if we're dealing with the action potential, now we no longer have an end plate potential, which means it was limited and it could not reach those voltage gated sodium and then potassium channels. The action potential means it reached threshold, and yes, we can get to that point. Okay. So what happens? We can get sodium entering the cell and potassium exiting the cell, and they both play a critical role in the action potential by rapidly, and rapidly it's important, the sodium entrance depolarizes, the potassium exit repolarizes. Remember the relationship of how many sodiums to how many potassiums, because that reinforces that 3-to-2 relationship thing we've been seeing a lot of recently. And look at the structures that are there. The terminal cisternae. What is a T-tubule? What is a triad? T-tubule is part of the sarcolemma. Terminal cisternae are part of the sarcoplasmic reticulum. The voltage-gated calcium channels that allow the calcium, which is intracellular in the case of skeletal muscle, unlike smooth muscle, which is extracellular, mostly, even though there's some intracellular. And so, how does the impulse moves along the T-tubule? The cisternae really don't move per se. What happens, they're just storage facilities, as a lot of smooth endoplasmic reticulum is. And so we either have stuff coming in or stuff going out, basically. So they're either storing the calcium or they're releasing the calcium, in this case, to the intracellular environment. Interestingly, look what happens when a muscle contracts. Take a look at that M line and H zone area. I think it's important. I guess and so I think as long as we keep putting additional calcium in, we're going to effectively, if you think of it that way, over time, we're going to have greater and more frequent contractions. Okay. And remember, be wary of the waste products. You should be very familiar with them for that anaerobic activity because you know your way around glycolysis very well. You know remember where and how the mechanism to make that lactic acid back into pyruvic acid. I would think you'd have covered out most of this in previous courses. I'm not remember which one of those many courses that you had. Okay. Don't forget that 80 to 120 golden zone, so to speak. What happens if you overstretch or you don't? If a muscle starts too contracted or too stretched out, basically why it can't generate power. Either it can't shorten enough or there aren't enough myosin heads available. I mean, all of those are significant because I really was fond of looking at that. Let's see. And remember, let's see, all the different roles of actin and all the different roles of myosin, the distinction between the heads, the whole pivoting apparatus. I think that's significant. That's there. Remember the location for the voltage gated channels and ligate, wherever all the voltage gated channels, whether it's for calcium, sodium, or potassium, where they're located, either in the nerve or the muscle fiber, and also where the ligand, gated channels are. And now we get mostly, we're sort of now, I think we're pretty well done with the muscle. Something came to mind. Oh, smooth muscle. That's why it came to mind. So smooth muscle. Remember the ratio again of thin to thick, why it appears to be smooth. Remember the distinction. Okay, extracellular calcium, not intracellular. It utilizes both. Does it have T tubules? Does it have troponin? Does it have tropomyosin? Okay, what kind of action? What kind of neurotransmitters might be used? So, and effectively, is there a neuromuscular conjunction? And if so, what neurotransmitters you use? Remember some of the terminology. Those caveolae. Understand the the calmodulin, the myosin kinases, the roles that they play that are there. Understand the relationship in the intermediate filaments, those love analogous or similar structures, like those various things that you will see. So, what happens to the calcium when the events trigger its release? Is it coming from the outside or the inside? Or if so, which one of those is most important? What specifically does it do? You start looking at the role of calmodulin. What specifically is activating the myosin kinase? What's there? What's a dense body? What's that neat arrangement it has? That zigzag pattern that you began to, and this one's a little bit different because it's more of a lattice work pattern that's there. The fact that it has the capability, what does its shape change? Does it effectively like a sarcomere? Does it shorten or does it move in some other direction? Is it more powerful? Is it less powerful? Is it more about endurance? All of those things are significant. So I suggest you take a goodly look at those that are there. And then we move back and we finally finish up the nervous system. For all of this has been going on. So you can see, I think muscle probably we have more about that than almost anything else. I think. As I'm scrolling through as I'm listening to this, and we're not that far away from being done. All the different glial cells, the four glial cells of the central nervous system, and their general roles. The two glial cells of the peripheral nervous system, so the equivalent, the Schwann cells, and their similarity to the oligodendrocytes and the fact they make myelin and the role of myelin, and not forgetting that they're different because the Schwann cells are single over a very long typical axon. And because of space limitations, the oligodendrocyte can bond up to 60 individual axons because you really don't have a lot of space. They're similar because they're wrapping their membrane, which is effectively the myelin, this neat kind of protein and lipid combination, which is basically what cell membranes are about. The myriad of roles of the astrocytes, understanding the blood-brain barrier, the role that both tight junctions, perhaps most importantly in the unique capillaries, and the role that the oligodendrocytes play. And don't forget the other ones that are there. The satellite cells we're really not sure of. The microglia are certainly important. The ependymal cells will play a role, particularly as we begin to look at cerebrospinal fluid. And we've looked at that a bit. We've looked at that more previously in anatomy, but certainly that's where they play a significant role. The different kinds of cells, those are the names. Okay, understanding the neuron is certainly going to be important. And getting more into that, just let me scroll down just a little bit. What creates that directionality? Remember, muscle and nerve are different. I spent a lot of time on that. What is the significant feature that ensures one-way flow of the impulse? We're not talking the movement of materials to and from, but specifically that one-way flow of the impulse, the idea of what's resting membrane potential, what plays a role in that. Okay, that's there. Why do they fire so rapidly? What seems to be the major portion? So they compared to uh, with that comparatively small absolute refractory period, that's really very significant. So if you don't know, I suggest you give it a good look and understand why what's going on. And again, how do we determine intensity? Okay, understand that. Okay, what effectively is an action potential? How do we describe it? How does it differ from those graded potentials? Remember, we have either excitatory ones or inhibitory ones. So all of those things, and why something would be, why would something be excited? We classify them as depolarizing or hyperpolarizing. So in the nervous system, and then we use the term depolarizing maybe a little bit differently in muscle than we do nerve. So make sure you understand what system it is, understand about different kinds of potentials that are there. Let's see what else. Covered that. Yeah. Again, I have several questions on resting membrane potential. Understand the role that proteins play internally, chloride plays externally, the relationship where the sodium is, where the potassium is. I'm sure you always know the leakage channels, etc. And maybe and the question that students typically find more difficult than others, we do a sequence question. It's five. Basically, it's a 5 point question. So basically, a sequence of event. What happens when a nerve fires? In order. Well, sure. I mean, think of what happens when nerve talks to nerve. An impulse comes from one nerve, right? And what that impulse does is it causes, when it goes to the terminal end of what we call the because we did join a synapse for our purposes in this. We have the presynaptic nerve. The first thing that has to happen, it has to trigger, that impulse has to trigger a voltage gated calcium channel. Mhm. At that point, it releases neurotransmitter, and it's assuming it's sufficient, an action potential will develop. Once the action potential, action, bigger part, once the action potential occurs or develops, what basically happens after that? We begin to see next voltage gated sodium channels, followed by voltage gated potassium channels in their activity. And lastly, we're going to see everything reproduce the interior. Remember, the sodium channels and potassium channels, those are ionic changes. Okay. And those ionic changes, so basically the sodium channels will create that wave of extraordinarily measurable depolarization, and then the potassium will repolarize it. Following that, okay, we have that, and now we've resolved the charge issue. Now we have to deal with restore the ionic conditions. That's what the sodium potassium pumps are all about. So, and I think that pretty much covers everything I wanted to say to you. If I think of anything else that might be on there, if I tweak it a little bit more, I'll certainly let you know, but I don't anticipate I'll do that. So thanks very much. Be seeing you tomorrow when we will continue and basically to all the neurotransmitters we haven't covered. Bye bye.